And good afternoon, fellow forensic scientists. So, as promised, this is a discussion about some DNA-based analyses, and there's a lot we can go through, and we'll see how much we do. I'll also throw up some PowerPoint stuff. Not all of the PowerPoint stuff that I'll throw up is actually stuff that we will cover, but I'm going to kind of hit the high points here, and may even actually transition to a PowerPoint. It's something I actually found on the web. But DNA is actually what I do, and so we've been actually doing a little bit of DNA analysis that you might do for forensics in the lab this semester, and we have some interesting results from our own. So here's the base. Everybody's got DNA. Now, within the population of all humans, we pretty much are identical at the nucleotide level. It's one of the things I think about in terms of the way we think about each other and look at each other, that the degree of similarity we have is gigantic compared to the little tiny differences. Obviously, if you look around the room, well, whatever is who's in your room, these differences between individuals end up being very, very subtle differences. Generally, what are called single nucleotide differences, or single nucleotide polymorphisms, or SNPs, that result in having curly hair, being tall, being short, the way your face looks. It's all controlled by the genes in your genome. And honestly, I would imagine that if you probably took say 100 different genes and mapped out the single nucleotide differences within the population, and then correlated that to facial, to appearance, or to skin tone, to hair texture, you probably, with around 100 genes, be able to have a pretty good way of identifying people. And we don't have that level of correlation and decoding, as it were. If you've ever seen the movie Gattaca, which is an interesting film, in which people would take a drop of DNA, and within moments they had your entire genome sequenced, and then, sorry, I'm looking at myself in the video and I keep on flipping my hair. Regardless, they were able to generate a facial image from your DNA. Now, theoretically, that is possible because I look the way I look. I look a little bit like my Uncle Herman, not because we stood next to each other while I was growing up, but because we share certain nucleotide differences in probably a very small set of genes that are responsible for the growth of your nose, how far your cheeks grow, the growth of your chin. Very subtle differences that result in all these different facial types and body types and characteristics that we see. So there's some very interesting things you can do with genomics that way. And if you've ever done a 23andMe thing, or if you know these things, where you spit into a tube, you hand the DNA off to the company, and they now have your DNA, and they're able to carry out a whole range of identifications of these single nucleotide changes that are associated with different things. Everything from the way cilantro might taste to you. And then they're constantly using the data set they have and asking those people that submitted various questions. How things taste, your preference in music, who knows what. And then they're trying to correlate this with those single nucleotide changes. So there's a lot of information in the genome that really is still being figured out. So we can have the genome sequenced, but to understand really what it means is a much more complicated process. All right, so in terms of identifying individuals based on their DNA, looking at the whole genome really doesn't work because we are so similar. But there are spots in the genome that are, in fact, not all that well conserved within individual members of the population. Most of our genome is quite conserved. So Trying to identify an individual based on their DNA is, in fact, a pretty clever trick. It is not just falling off a log. Because if you were to look at the genes that are used to generate proteins, there really isn't that much difference. And it would be difficult to actually have a really good, sort of beyond the shadow of a doubt, court level, law level required establishment that this DNA came from this individual. So as people began studying the genome, these different patches in the genome became apparent that contain what are known as repeat sequences. And specifically, there are regions in the genome that contain what are known as short, i.e., 4 to 6 nucleotides, tandem, meaning one after the other, repeats. And so these are referred to as STRs, short tandem repeats. And it may be something like GATC. And this 4 nucleotide repeat can be repeated over and over and over and over again. And depending on the short tandem repeat site, within the population, you might have 3 repeats, and then you find people that will have anywhere from 3 to say 18 repeats. And these are going to be spots of the genome that really don't do anything. And so they're not going to be selected for or against because, one might make you a little healthier or one might make you a little more sensitive to diseases. No, they're just going to be sitting around. Now, back in the 90s, once more of the genome had been sequenced and the presence of these short tandem repeats was identified, people began working on using them as an identification tool. And the sites on the chromosomes where these STRs happen to be, and there's a whole bunch of them, these are referred to as loci, or individual as a locus. And so you have what are known as short tandem repeat loci, or locations within the genome. Every chromosome has got bunches of STR sites. Now, what makes these things work is that outside of the region of repeat, we're going to have sequences of DNA that are absolutely invariant within the population. And that's important because that gives us a uniform tool, a sequence, that we can use to actually pull out this STR. All right, so the next bit of information we need to know is a way we can manipulate DNA and how we can actually take and identify these STR loci and actually study them. Because you take DNA and it is just sequence. What we have to do is essentially amplify up this very narrow spot. We got to generate a lot of DNA from just that region. And prior to 85 or so, this was a bit of a difficult trick. But a technology came about that makes it so this is in fact really pretty easy. It's a technology known as the polymerase chain reaction, better known as PCR. PCR is such a critical tool in molecular biology and DNA analysis. We do it pretty much every day. We've got multiple PCR machines. This is the way DNA is analyzed these days. And in principle, it's really quite easy. What you have is you got to have a little bit of sequence of the genome. And so you've got a site in the genome that you're interested in. Now, it's double-stranded DNA, but that's not really all that critical for our discussion. What we have then is, let's just say that we got a region that contains short tandem repeats here. And you can synthesize and purchase something called an oligonucleotide primer. This is a DNA sequence that will base pair or hybridize to this side of the STR and to this side of the STR. That is what's known in the business as the flanking regions on either side of it. Then you add the sample DNA, the template DNA. You add what are known, these two sequences, these things that you buy, they're known as primers. And you put it into the polymerase chain reaction machine. It's basically like a DNA duplicating machine. And you allow it to run. It goes through these cycles, and you'll usually do 30 to 35, 40 cycles. And what you do is you'll generate very large amounts of just this piece of DNA. Now, the next step then is to determine the number of repeats that are between those two primers. And you do that by the size. So this comes down to counting nucleotides, counting the individual bases. And let's just take, for an example, a tetranucleotide repeat like we just had, GATC. So if you've got 10 repeats, that will be this times 10, that'll be 40 nucleotides. All right? If you've got 11 repeats, that'll be 44 nucleotides. So what we can then do, once we've amplified up this piece of DNA, is we can size it. Now, this actually is where a little bit of equipment comes into play. The standard tool for sizing these things is something known as capillary electrophoresis. And we don't have a capillary electrophoresis machine here. We have what is known as gel electrophoresis, where we run the DNA through this gel material, and the bands or the fragments separate out based on their size. Smaller stuff runs faster, slower stuff runs slower. And you can separate the samples out. And actually, I'll probably show you some pictures here in just a moment of some of the work we've been doing. Well, what you can do is size and determine the number of repeats at a specific spot. And this becomes the basis for what is now known as CODIS. So back, I think in 96, 93, an international group of scientists gathered, and they defined what would be the standard set of CODIS alleles. And I think the current forensic STR system is looking at 16 CODIS allele, or actually that's a new term, I don't want to use that term yet, 16 CODIS loci, 16 spots in the genome. These are the industry standards for the CODIS database. And it works really well, particularly compared to like fingerprints, where with fingerprinting, as we were playing with this last week, you got whorls, you got swoops, you got squiggles. Trying to define a uniform coding system for this that you can put into a database is pretty hard. Let's think of an Excel spreadsheet. With CODIS, though, it's really easy because you've got like a CODIS loci that might be called D3. And within that, there are a set of repeat numbers within this D3 locus that are found in the population. And so, say you've got 7 of these repeats. So you'd be marked as being D3 7. And then you might have D18. And so you got 6 there. Now, another point behind this is that because you have one chromosome from mom, one chromosome from dad, quite likely the mom chromosome will have one number of repeats, the dad chromosome will have a different number of repeats, and so you end up actually most of the time with 2 numbers. So it would be D3, 7, and 9, and D18, 6, and 8. I'm just making these up. All right, this becomes a really easy database kind of identifying system. And you establish this for all 16 CODIS loci. Now, you do this, now in our lab, we do this one at a time. In the forensics labs, state forensics labs, you can do something called a multiplex, where you can put in a number of primer sets for the different kinds of CODIS loci and pull out all these different kinds of CODIS data all in one throw. We just don't do that. And what you can end up with then is a really clever little data set that becomes a unique identifier, because the next step is this. Each one of these loci are going to have a set number of repeats seen in the population. So the repeat number becomes something we refer to as an allele, which is essentially the flavor. So with more standard genes, you might have an allele for curly hair and an allele for straight hair. You might have an allele for red eye, or not red eyes, hopefully not, blue eyes versus brown eyes. And so alleles are the kind of, you can think of as the flavors are the things that are exhibited by that specific gene. So you have one gene, and one gene can have multiple alleles, multiple flavors. Well, at each of these 16 loci, there can be a whole range of repeat numbers, and that's the number of alleles. So the number of alleles gets to be pretty big. And then when you begin combining that, man, the statistical ability to distinguish individuals is staggering. i.e., when you say that this DNA came from this dude, that is a lead pipe cinch. You cannot argue with it. It is the thing. Now, from a defense perspective, about the only thing you can do is ask, all right, how was the evidence handled? Do you have a good, clear chain of custody? The defense is going to want to take some of that DNA and run the PCR and the analysis themselves. Do you have any more of it? Oh, no, we had just one little bit and we used it all up. That's not great, because how do we know you didn't fudge it? You didn't jack with the data. Well, we would never do that. Well, you can go back and forth, but the actual DNA evidence itself, that's rock solid, man. You can't jack with that. And we'll see here in a moment, there are some very cool statistical charts that look at allele frequency and the probability that somebody might have this allele. That is, out of the population, how many people have this. You start tallying those up, and it gets to be the number of people that it would take in order to have more than one person with this combination. It gets to be into the trillions really fast. That means that there is a 1 in 10 trillion chance that somebody else would have the same CODIS profile as our defendant. How are you going to argue against that? Pretty quickly with CODIS, if you have a standard CODIS run, you've got a statistical likelihood of a match would require more people, more humans than have ever been on the planet in the history of human civilization. So, sorry, Charlie, nah. If it's you, it's you. One of the first times that DNA evidence was used in a high-profile case came with the O.J. Simpson trial back in the 90s. The trial finished up, I think, in 93, 94. Mr. Simpson's wife was found murdered along with her boyfriend, and there was, in the area around it, a glove that had blood on it, and they were able, forensic investigators were able to identify the different blood from the victims and identify blood from a third person that turned out to be O.J. Simpson, Nicole Simpson's former husband. And the accuracy of the PCR test and of the CODIS analysis was absolutely unassailable. It was his blood. The question became, how did the blood get there? And where was all the blood that was taken from Mr. Simpson right after his arrest? Several vials of blood were drawn for a DNA analysis. Now we don't do blood, we use mouth swabs. But blood was taken, and the question then became from the defense is, can you account for every single drop of that blood, with the notion that maybe they planted blood onto the glove? So the defense was able to establish sufficient doubt that while, yes, the blood was from Mr. Simpson, how did the blood get there? There was sufficient doubt. It's a mess, and what do you want to say? But that is, about the only out you're going to have in a defense of DNA analysis is that, can you make sure that the sample was handled properly, and there's a chain of evidence and chain of custody that makes sure this stuff has the appropriate provenance. Now, by not taking blood, but by taking a mouth swab, it'd be a little bit harder to contaminate the sample. But it's a game. Now, there are a number of difficulties because identifying clean sample from a crime scene is a lot harder than identifying a clean sample from an individual. For an individual, you spit into a tube, that'll do it. You take a little brush, wash, scrub the inside of their mouth. You can get cells that way, that'll do it. You can certainly draw blood. We can get very clean DNA from individuals very simply. From a crime scene, that's going to be a lot more difficult. First off, a huge number of DNA-based evidence questions are involved in rape cases. And it makes me kind of like, oh damn, that is just scary sad, in that there are tools and techniques that can separate sperm from the epithelial cells of the woman, of the victim. And you can then begin to actually separate things out that way. Just as a twist, before I go into some of the PowerPoints, just to look at some of the diagrams, one of the new things that's coming along is not just doing CODIS analysis, and sometimes if an individual is not in the CODIS database, then you don't have a direct lead. But you can also identify other spots in the genome, and you can tell, not necessarily to exclusion, but you could say, okay, the victim or the DNA came from somebody who is Hispanic, somebody who has brown hair, a whole range of different kinds of physical characteristics. As I say, we don't really have the ability to generate a face, but that'd be a pretty cool thing to be able to do. And I would say, I would bet in the next 20 years, we probably will be able to do that. So, short of having undergone, facial reconstruction surgery, and you wouldn't necessarily be able to tell if somebody has a beard or not, you'd be able to get a picture that, you could probably recognize somebody in the airport that way. Well, that's another way you can use the DNA evidence, but that's not going to generate the degree of exclusivity that the CODIS stuff does. Let's see, what time is it here? I'm going to go through a couple of figures just on this PowerPoint and then give you a basic exercise in this and see where this goes. All right, so let me get the slide projector going. So here is a little bit of a PowerPoint thing on DNA analysis. This first image is showing the current forensic STR mixes, where it's known as the multiplex. In the trade, it is known as the PowerPlex 16, and you can just actually buy this commercially. This is going to be the DNA sequences required to amplify up the 16 different CODIS alleles. So they have a range of sizes that the alleles will be, and I'm going to kill a little bit more light. Hold on a second. Yeah, easier to see. At the same time, I'm going to give you the slides, so you'll see me as this ghost. Hi. Is this working? Oh, what the hell? We'll go through it anyway. All right. Nah, hold on a second. I'm going to turn on a little bit of light. All right, so this is an example of the results from capillary electrophoresis, and you end up with these little peaks. So this is a sizing standard, and one of the things that is critical is something called an allelic ladder, which is a mix of all the different kinds of numbers that a specific CODIS loci might give you, and that's useful for being able to define exactly the number of repeats. So it ends up being a little bit complicated. What I'm going to show you, and our exercise won't be this complicated. But you can then do a series of CODIS loci. Now, let's just look at some of the numbers here. The hypothetical likelihood of occurrence. One locus, you have a 1 in 18 chance. Two loci, because one thing, if you know your probability and your statistics, what you're doing is you've got the number of possibilities, say it's 18, and to have any one of those numbers along with any one of the numbers in the second locus is not just additive, but it's actually going to be multiplicative. So you've got a 1 in 18 chance, and then if the next one is also a 1 in 18 chance, it's 18 times 18, which I think is 360. So you start to multiply, and baby, it gets to be a big number in a hurry. So 1 locus, 1 in 18, 2, 1, 360, 3, 1 in 18,000, 4, 1 in 79,000, or no, 7,792,000. Yeah. All right. Yeah. 18,000? No. That's, yeah. I'm getting lost. Regardless. Oh, yeah, they went that way. So you could theoretically then, with 3 loci, that's 18,000 people. With 4 loci, you go from 18,000 up to 792,000. That's more people that are in downstate Illinois at that point, and so you really could have a pretty good discriminatory bit of evidence there. But if you keep on going, check this out. 9, you got 1 in 10 to the 10th, which is a big ass number. 16, you got 1 in 10 to the 17th. At the moment, the current world population, 6.3 billion. It's actually more than that, 7 billion. But that's 1 times, or 7 times 10 to the 8th. So you're talking a lot more people it would take in order to have the possibility or the likely probability of having 2 people with the same genotype. So now this works really, really well. Now, in order to do the data analysis, one of the things you have to have is a series of what we call the business controls. That it would be a negative control that doesn't have any DNA in it, and then you compare it with these locus-specific known sequences, and that allows you to get a range of sizes, and you can precisely size yours. I'll just say from my own personal experience, getting a negative control is not nearly as easy as you might think. That is, running a sample that has no DNA, you've got to be really careful. There is human DNA floating around everywhere. You touch something, and you're able to get a little bit of DNA off of it. I haven't done like sort of intentionally trying to get DNA off of a fingerprint, and I'm not sure how well it would work, but I do know that just casual handling of material, you do end up transferring some DNA onto it. Yeah, so these are some of the things you can do with this. Obviously, forensics cases, but it's also used for paternity testing. There is a database now of CODIS alleles that pretty much everybody that's a convicted felon has to donate a little bit of their DNA. It's run through. It's useful for missing persons, mass disasters, all kinds of things. A military DNA dog tag. Anybody in the active military probably has had a CODIS analysis done of them, just in case for purposes of identifying remains. All right, so another thing that's been done, or that's happened recently, is that because a lot of people have been donating their DNA voluntarily to places like 23andMe and other kinds of genome analysis, Ancestry.com, the police actually can scan the data there and look for not CODIS matches, but other kinds of links, and they can actually identify familial, somebody's brother or sister, or mother or father, or daughter, and son. They can be able to track that way. There was a court case last year about the use of these DNA databases. Now I think if you go into 23andMe, you have to opt in to allowing your DNA to be used in some kind of criminal investigation. So this is what an STR profile might look like. And let's see if there's anything here. But beep. Yep, snaps and processing. One of the things that we'll be talking with my friend Sarah next week, DNA quantitation is something that's required. Forensic labs are required by law to quantitate the amount of human DNA in a crime scene sample. And she was saying that a quant stop happens a lot. That is, they just didn't get enough DNA to actually get a good sample. So the labs themselves are fairly carefully regulated. The FBI has got standards for CODIS labs, and there are a whole set of just sort of standard lab rules and things. And there you go. So this is what the PowerPlex 16 might look like. And you have then these individual lines are the allelic ladders for the different spots. You just count up and you assign the number that way. Now, I'll let you look at the figure in the text, all right, when I send it to you, but this is the allele frequency data set. And it looks at the allele frequency within Caucasians, African Americans, Hispanic Americans, and the allele frequencies as we break it down. You end up having a pretty good set of statistics that allow you to actually match and agree to to a level at a court required level of discrimination. All right, so that's what we have to say about some basic DNA stuff. I'm going to get a little worksheet out to you as well that looks at some STRs. You'll identify the repeats and the numbers. We'll go over that next week as well. Let's see, on Monday, I'm not sure, Sarah is working on a recording maybe. She's going to record her seminar. I'm not sure if she's going to be able to zoom in live with us or not, but we'll have that to look at and talk about, and we'll see where we go from there. All right, thanks guys for your attention. I'm Dr. G. Ooh, just for the heck of it, I'm going to show you some of the stuff that we've done specifically this semester. Lucy Koger and I working on some DNA work, and let's see. At this point you might have turned off, and that's fine too. So this is what CODIS looks like on our systems, and we haven't run the gel very far here. When we run it further, we end up getting individual bands. And so here's two different bands. This is for what is called D5, and this is D16. And when we run them out a little bit further, we actually are seeing two bands. The difficulty Lucy and I are running into is sizing them. We haven't been able to get a exact number, and this is actually one of the figures from our latest work. We got a couple of them around, but anyway, theoretically, we would like to see differences in the sizes of these two bands. That we don't see great differences makes me kind of go, ooh, this could be artifact. I don't know. We should be seeing more differences than this. What this means, though, is, on one hand, that our gel separation technology isn't great enough to distinguish between something that's got 10 4-nucleotide repeats and something that has 11 4-nucleotide repeats. Anyway, that's what we have to show for ourselves. I really want a capillary electrophoresis machine, but let's see if we get one. Hey, that's all I know for now. Thanks, and I will check in with you cats later.